Well, thank you so much for having me today. I won't be able to read through the chat while I am presenting. And as I noted, I'm having some difficulty hearing, but I will happily come back to the chat questions as we go through the presentation. So today, first, I want to give thanks to our colleagues and partners at Philly Korea for hosting me for this presentation to present to you on industrialized CRISPR and applications in disease research and therapeutics. So my name is Rachel Yanas. I am the Director of North American Sales, as well as managing our distribution network, largely through Asia Pacific. So I'm pleased to be presenting to you today and hopeful that I'm able to share some new insights with you. So first, what I would like to do as we get started is go over some of the details of what we will be covering. And I would like to give a brief introduction to CRISPR. We will talk through some abilities to achieve reproducible gene editing in many diverse cell types and how we are able to accomplish and support this, as well as cover how we can accelerate disease research and enable large-scale studies with industrialized CRISPR. And then we'll certainly wrap up with closing and question and answers. So as I proceed, this was actually supposed to be an animation. And what I wanted to share with you is some numbers, 4.3, 171, and 5 as we get started. And this is really to help set the stage for this conversation. So as I was doing some of my research into the Korean market, I discovered a few interesting facts that most notably, 4.3% was identified as the amount of GDP that South Korea spent annually on research and development that was reported in 2019. So this was a fact from about 3 years ago. And this is striking as this is more than both the United States and China at that time. So that there's very much a heavy investment in research within South Korea. And notably, I discovered that there are 171 estimated number of biotech and pharmaceutical companies within South Korea as well. So to go along with that investment of research dollars, it is being funded not only into the large number of academic centers, but also certainly into industry. And then perhaps most notably and most recent that's come to light is that I discovered there were 5 new drugs that were both developed in Korea and approved by South Korea's Ministry of Food and Drug Safety, or the MFDS, in 2021. So for me, and I think for everyone on this call, while these may not be new facts, they are certainly very much in support of why it's helpful for us to chat about new technologies that can continue to accelerate the growth of the biotech and pharma company within South Korea, and how both Synthago and Philly Korea are enabling that through CRISPR gene editing methods. So as we move on, I wanted to, before going into the details, speak to a story about a small biotech company in the United States that I think is a good example for everyone that works in research and really in drug discovery, early stage of research in academia, who is looking to publish, to meet strict timelines, to satisfy investment boards, and founders of companies. So this company, whose name I'm not able to disclose, but I can tell you about their background and what they were trying to accomplish. This company was funded by venture capital. So they have very time-sensitive investors with board meetings at specific quarterly dates. And the company itself was targeting areas of protein degradation, and they had several targets in mind to investigate for their drug target. But they needed to have or discover a model that would demonstrate the drug's mechanism of action. And they only had about 3 months time to do this. Now, this is not a new story for many companies that exist, but it's one that those of you working in biotech and pharma may be able to sympathize and understand urgent timelines, whether they are from your own manager or your PI or board that your company has to report to. So this firm or this biotech account, they had identified what they thought would be the best gene target for them to study and really demonstrate the mechanism of action of their drug candidate. They didn't think they had time to expand into other models, looking at other targets that may be the right gene target for this drug. So they decided they wanted to take a bet and pursue just one target, creating a single cell clonal line either in A549 cells or HEC293s in order to meet timelines in which they needed to present data to their board. Now, in many cases, they may have done just this, but it would have introduced a lot of risk in meeting their board deadlines and the data generation that was required to go on to the next step of their process. So they were fortunate to come across an organization that proposed an alternative solution to this. Rather than invest in just a single model and a single gene target, which they felt they were restricted to do so by resources, they were presented with an alternative solution in which they could target not just one gene, but 4 genes of interest, and not in just one single cell line, but across 3 different cell lines, and still be able to do this and achieve their goals of presenting data to their board at their meeting within March. And they were able to do this by generating not what we would call single cell clones of these gene knockout models, but by generating knockout cell pools in which a large majority of the population has the gene target knocked out, that's at a level high enough in which they would be able to use these models for validation experiments and identify top candidates for their gene of interest. So presented with this option, the team at this company decided to pursue this route. So rather than simply generate one model, they moved forward with 12. And this was a wonderful decision on their part because they were able to leverage all 12 of these models and identify 2 distinct gene targets that were actually different from their original target of interest. So had they gone with their initial plan, they wouldn't have had the right data or the right target to present to their board, and they would have had to start over again with a new cell line and a new target. So this approach, by targeting multiple genes at one time across different cell lines and really leveraging what we call CRISPR at scale, they were able to get the data that they need, present to their board, and ultimately this led to successful rounds of additional funding and ultimately an IPO for this organization. So what this CRISPR at scale that they were able to take advantage of really enabled them to do was build better experiments by obtaining actionable data, leveraging knockout cell pools that were delivered to them in just 1 month's time frame, and then subsequently able to run their assays of interest. Because these knockout cell pools were made through automated measures, they could have confidence in the process and the product that was delivered to them, and they were able to complete all of this within just 3 months' time, running 12 different cell lines in parallel. And so Synthego, at the center of this story, providing this option to the customer, was able to deliver a wide set of cell models such that they could run their assays in parallel and advance 2 new gene targets in time for their board meeting. So I present this as a learning experience to keep in mind how you might consider leveraging or using CRISPR gene editing tools at scale to advance and accelerate your research programs with confidence in the outcomes that you will have strong data to support your goals. So now we'll rewind a little bit and cover what is CRISPR, in just a few quick slides, because certainly CRISPR is not a novel technology, but what CRISPR really is now, having been published on 10 years ago, we've come to view it as a programmable genome rewriting tool. In essence, or at our foundation, when we consider this phrase, we know that the human genome was first fully published on and identified about 20 years ago. So really not that long ago. And CRISPR itself was then published on 10 years ago in 2012. Prior to where we are at today, next generation sequencing has also developed and enabled us to truly read and fully understand large genomes in great depth, allowing us to read the genome as we would read a code. CRISPR, combined with that ability to read the genome, allows us now to rewrite the genome by leveraging these two components, the Cas9 nuclease that we see in this gray component and our guide RNAs. So how does this programmable genome writing work? So we know that CRISPR-Cas9 is a simple but very elegant system. It is combined of the Cas9 protein or endonuclease that doesn't change. Cas9 itself can be used no matter if you are editing an immortalized cell line, if you're editing a human genome, a mouse genome, or a zebrafish, and many other different species can be utilized with the same Cas9 protein, which is one of the truly elegant and simple components about this that I quite love and appreciate. So in addition to this Cas9 protein, we also utilize our sgRNA or single guide RNA. And this is the powerful but small 100 base pair RNA sequence that is designed to target a very specific and precise region of the genome. Now, when these two components are combined to create a ribonucleoprotein, they are then transfected into the model of interest or the cell of interest, and they locate that genome sequence that we would like to edit with the guide RNA, and the Cas9 nuclease then creates a double-stranded break. Now, this is really all that the CRISPR-Cas9 system does, is create this double-stranded break. The subsequent actions then are the result of cellular repair. Now, most frequently, we see non-homologous end joining occur within our DNA, where the cell or the DNA strand is hurrying to recover that double-stranded break. This is not healthy for our population, and ultimately this can yield random insertions and deletions into the genome and ultimately create a functional protein knockout if we introduce a frame shift mutation or cause a protein truncation. Now, the other method of repair is homology-directed repair, in which a DNA template must be provided to insert into that double-stranded location. Now, this occurs much less frequently than non-homologous end joining because of the need to insert that additional piece of DNA, and because of the precise nature of having that DNA sequence combined without any other insertions, deletions, or changes to the sequence. But homology-directed repair, as opposed to allowing us to study loss of function, allows us to create single nucleotide variants or point mutations that may be associated with specific diseases such as Alzheimer's or Huntington's. Knock-ins also allow us to insert tags so we can look at protein degradation with things like HiBET or nanoluciferases to look at fluorescence expressions with the GFPs. So there are a number of different DNA repair templates that we can introduce, and how we can leverage homology-directed repair to study different mechanisms in addition to the functionality of specific point mutations. So that's really the basic foundation of CRISPR-Cas9 and how it works in our cells. And it really is a phenomenal tool because of that simplicity and what it unlocks for the potential of studying different genomes and disease states. However, we've found that the promise of CRISPR still remains somewhat unfulfilled. We did perform a survey amongst numerous different researchers who are trying to use CRISPR themselves to create their own models, performing their own editing and transfection. And what we found is that many researchers indicate that performing CRISPR gene editing is challenging for themselves. They're only able to generate on average 43% editing efficiency, which is an average is quite low. And we find that this becomes even lower when we look at creating knock-ins or that homology-directed repair. Additionally, it takes them roughly 7 attempts to achieve a successful CRISPR-edited cell line, and this can take up to 100 hours, if not more, per cell model that they're trying to generate. So as a result of this, most researchers have indicated that they are unsatisfied with their results. Only 15% surveyed were happy or pleased with what they were able to accomplish. And what I think is even most striking in this case is that researchers are studying on average 7 targets when realistically they would like to study at least 17 different targets for their research goals. So we know that disease biology requires us to investigate hundreds of millions of different genes or variants or mutations within those genes. But because of this cumbersome process, or what's become cumbersome for researchers to do, it's not successful, or we're not able to unlock this full potential. Because ultimately, with CRISPR and creating engineered cell models, ultimately you're making a tool for your far more interesting research and therapy developments. So we find that it's clear researchers need a better, predictable, and scalable way to obtain the models for their work that leverage or require gene editing tools or gene edited tools. So what I would like to speak to today then is the platforms that Synthigo has developed that enable this scale, that remove those barriers to studying hundreds of combinations of genes and variants and mutations, so that you don't have to limit yourself to just studying 1 to 2 targets, but can expand your entire research scope to study 10, 20, 50, or hundreds of targets pertinent to your focal area. So Synthigo is as we know, is a genome engineering platform company that has about 450 people within the organization. And I highlight the number of people because it's important to recognize that those people are all involved in these different technologies. We have chemists who are involved in our RNA synthesis or the production of guide RNAs that are used in CRISPR, and who are trying to identify novel chemistries for new methods of CRISPR engineering. We have computer programmers who help build our softwares that supply us with machine learning programs so that we can better improve our editing capabilities, our guide design processes, and our strategies. And then we have really hardware engineers that build our automation, they build our instrumentation that is customized for our industrialized CRISPR capabilities. And of course, we have cell biologists and molecular biologists who are really implementing novel genome editing strategies in our research groups, as well as performing and overseeing the high throughput CRISPR editing work that we do for researchers globally. So all of these technologies and skills that our team has feed into the 2 platforms that we've created. Halo, which is our platform for producing synthetic chemically modified guide RNAs, or our CRISPR tools as we call them, and our Eclipse platform, which creates our engineered cell models. So these 2 platforms then provide solutions for of all spectrums of research and drug and therapeutic discovery and development, from target identification and validation through disease modeling and research to cell and gene therapy engineering or manufacturing. And this just indicates some of the different products that we offer off of our platforms for these various applications. So next, we will cover sort of what that Playload platform is in a bit greater detail and the various options that we have for synthetic single guide RNAs. So as I noted, HALO is the platform through which we produce or synthesize our guide RNAs that includes the combination of chemistry, of machine learning, so we understand how better to synthesize guides and make sequences, and of course automation so that we can produce not just one but thousands of guide RNAs at a time and throughout the course of a week. So all of these factors combine to produce very high quality standardized reagents through innovative and new processes that are proprietary to Syntheco's platform capabilities and enable discovery applications, whether that's through standard SP or S-pyogenase Cas9 or SA-Cas9 or other nucleases such as Cas12a that can be utilized in individual editing experiments. We've also leveraged our capabilities for synthesis to make arrayed loss of function screening libraries, which I will speak in more detail on. And then we also have clinical grade reagents that we make in our GMP facilities for the applications in cell and gene therapy development, some of which are in, I would say, preclinical or even early stage clinical trial phases. So when we look at the synthetic guide RNA, which is the foundation of our entire HALO platform, and also really the foundation of our cell engineering capabilities, want to highlight that what we are making and what we utilize is a completely synthetic RNA compared to what was historically the gold standard, or CRISPR plasmids, which would be transfected into models for creating gene editing cell lines or gene edited organisms. Now, there are several limitations we find with plasmids that often they can be cumbersome for sensitive cell lines, that they are constitutively expressed in certain cell lines as well, or in all cell lines really, which can lead to higher off-target effects. And as we can see from the data here, the plasmids actually did not perform nearly as well as our synthetic chemically modified guide RNAs. So if we look at our various titrations of plasmid in the gray bars versus our high-performing synthetic guides, it's clear to see that the indel frequency, or those patterns of random insertions and deletions, is much higher with a synthetic reagent. Furthermore, when we combine those synthetic guides with the Cas9 protein to form that ribonuclear protein, we find that we have much better viability post-transfection or post-nucleofection compared to plasmids, which, based on the size and the delivery mechanism, it can be very harsh for these cells. So this is particularly of importance as we look at editing challenging cell lines, some of our immune cell types, as well as primary cells such as T cells, hematopoietic progenitor cells, or other similar models. So two important factors to consider when you're looking at the source of the guide RNA and why we've really built our platforms off of synthetic reagents. Now, we've taken that further. In prior seminars, we've gone through much greater detail about the performance of an individual guide, how to use that, and how to design experiments. But what I'd really like to talk to more is our multi-guide approach that we use for loss of function studies, and really use for these large arrayed screens that allow you to look at the loss of function and the impact of that function on things such as morphology, general cell health, and things beyond simple viability. So what we have done is, with our bioinformatics team, we've built what we call a multi-guide design that yields a much better knockout for our gene target of interest. So this top chart here, if any of you are familiar with our ICE tool, which is a way to analyze the indel frequencies or the editing efficiency of a CRISPR edit, what we look at here is the contribution of different indels, those random insertions and deletions. Now, when we use a single guide target, which we know works quite well, what we often see is those single guides will yield in-frame mutations, or those in-frame indels that are going to be multiples of 3. And because that's in-frame, we're not likely to see a loss of function or that early introduction of a stop codon and protein truncation. So while these editing efficiencies are high, they don't necessarily guarantee loss of function. So what we have done is built a proprietary design where we actually combine 3 unique guides together in a specific and intentional design that causes them to work together to create large fragment deletions or dropouts of portions of the DNA. And when we do this, this will lead to a substantial disruption of the gene target of interest or the sequence, such that a loss of function or a truncated protein is created, and we will not see any function. So this design strategy has been implemented for a few years now in our gene knockout kits, as well as our arrayed loss of function screening libraries from small protein families all the way up through the whole human genome, as well as the whole mouse genome. And this just lists out or shows the number of gene targets that we've been able to design for. And you see that well over 90% of the whole human genome is able to leverage this multi-guide approach to create loss of function. So you might wonder, okay, does this really change the actual knockout, and how can we be sure that using 3 guides will lead to this true loss of function? So what we're looking at here is just a comparison of individual guides and indel frequency, or that, individual guides or the single guide RNAs compared against this multi-guide approach, or what's labeled as multiplexed here. And we're looking at the indel frequency, which does include those 3 base pair insertions or deletions, or the multiples of 3. And we compare that to what we call our knockout score, which is the indels that are not multiples of 3. So we remove those in-frame mutations. And it also includes large frame shifts of greater than 20 base pairs, regardless if those are in frame or not, because those large fragments are substantial enough that we would see loss of function, as I noted previously. So this knockout score is a much better indicator of true loss of function. Now, when we look at these compared together, the indel frequency doesn't differ much if we look at just the single guide versus the multiplex, but that's not the component we're really interested in. What we're looking at is the mean, or the average, of those individual guides for their knockout score, which is quite low, below 40%, compared to the median knockout score for our multi-guide approach, which is actually quite high and nearing 89% and close to 90%. So this is really what speaks to us and is a strong indicator and gives confidence that this approach will yield high loss of function in a pooled population where we will have some wild type or unedited cells, and a large population of those with the gene knocked out. And this design enables researchers to leverage this multi-guide format for studying large number of targets for loss of function. So what that has enabled us to do then is build these arrayed screening libraries for loss of function. Just to give a visual of what that looks like, we can build libraries within 384 well plates or 96 well plates also. And you can see the configuration where we're targeting gene X in well B2. And within that well, we will have those 3 distinct guides designed and combined to create that loss of function knockout of this target. So this format really enables you to have ready to transfect RNAs, to be able to create a master stop plate and then allocate or stamp out daughter plates that yield high knockout scores, strong confidence in your loss of function, strong confidence that you won't see false negatives, nor even false positives in your results. And because this is in a plated format versus what I'm sure many of you are aware of is a pooled library, the plated format really allows you to look at complex assays or readouts. So let's say you are working in neuroscience and you want to study the impact of a gene on, or multiple genes on neurite outgrowth. You can leverage that in microscopy-based assays within this plated format. You can also perform bioluminescence assays or CAS-based assays versus pooled screens where you can only simply look at live dead. So this format is very user-friendly and particularly important for working with primary cells and even some of those challenging cell types. So I want to then go through a few examples of how we've seen this multi-guide approach be leveraged. And in this case, first we're looking at the pilot work done by a group called the Weissman Lab out of the University of California in San Francisco, who was needed to leverage primary dendritic cells to look at immune responses to LPS stimulation. Now, before they could run a large screen and identify new targets or novel targets associated with this LPS response pathway, they wanted to ensure that they could yield high knockout scores or knockout efficiency and loss of function in their dendritic population, which can be very challenging to transfect and edit with CRISPR. So they used the multi-guide approach, and what we see here is when they compared against their individual guides and that multi-guide approach here, what they were able to observe is that the multi-guide indeed did deliver many large fragment deletions of greater than 50 nucleotides, as indicated by the key, compared to those single guides that were generally much smaller deletions present in the population. So this allowed for them to optimize, to gain confidence that they would see large fragment deletions, and perform or assess their results by NGS in addition to the assays that they would subsequently perform. Once they completed this pilot experiment, they then moved into a large-scale knockout screen or loss of function screen with their primary dendritic cells. So each of these individual bars represents a single well or a single knockout from 280 distinct gene targets that they were knocking down in a 384 well plate for their knockout screen. The orange bars are really indicative of those large fragment deletions. So you can see by and large that population had those present. And overall, they saw 97% is the median editing efficiency as assessed by next-gen sequencing of these 280 different gene targets. What they were able then to do is by running their LPS stimulation and response assay was identify two genetic factors out of this population that were potential contributors to the human immune response to this bacterial infection. Now they're able to take this information and continue to investigate and study these targets as a result of the large-scale screen they were able to do here, which would not have been possible with a pooled CRISPR screen and may not have been nearly as successful if they were using just individual guide RNAs for these knockouts. So I want to also highlight that, in this case, they simply received the plate of guide RNAs from us and performed the transfection themselves. But what we will see in this next example is actually going a step further. And I realize this has a lot of content on the slide, so I will walk through slowly. But what we were fortunate enough to do throughout the pandemic was partner with the lead investigator, Nevin Krogan, of a large collaboration for COVID-19 therapeutic development or discovery. So Nevin is a professor and director at the Gladstone Institute and the University of California, San Francisco. And much of his research does utilize bioinformatics information to identify these different protein-protein interaction maps to understand how proteins from different species, from different virus and host are working together. And in this case, coming out of or going really into the pandemic, Nevin focused his work on SARS-CoV-2 and was able to very quickly build this interaction map of the SARS-CoV-2 proteins and different human proteins that were interacting and working together. Now, he was able to take this data and partner with Synthico, or I should say we were fortunate to partner with him after building this map, and design guide RNA sequences in this multi-guide format to build what we now call the COVID Interactome library. So the Synthico team took the human proteins that he had identified from this map, designed our multi-guide approach for each of those proteins, which came out to about 400 unique genes that were targeted. And we built an arrayed library much in the same format as I showed you on the prior slide in 384 well plates or 96 well plate. But rather than simply deliver the guide RNAs to Nevin and his partners, we went one step further and we created the knockout or loss of function cell populations for him. We took the plate of those guide RNA libraries and we transected two different cell types. The lung epithelial cell A549 that was overexpressing ACE2+, which is a protein required for COVID-19 infection. And we also transfected the library into CACO2 cells, which are a gut epithelial cell line that is very readily and easily infected by COVID-19. So we were able to then create multiple cell libraries that had these 400 gene targets knocked out and ship them out to the various collaborators at Institut Pasteur, the Icahn School of Medicine at Mount Sinai, Georgia State University, and a few others. And we were able to do all of this within about 3 weeks' time. This then allowed Nevin and his team of collaborators to run a number of different assays in which they could identify various gene targets that were potentiators of COVID-19 and the infection process. After looking at those targets, they've then taken several of them into phase 3 studies within just 9 months time frame. And what they were doing in identifying these gene targets and taking them to trials was actually screening them against pre-approved or formally approved FDA drugs that would potentially be repurposed for COVID-19 treatment. So as we look at just a summary on this slide of what Nevin's team did, and how we were able to support them is we created over 300 distinct models in both of those cell lines, the A549 and the CACO2, delivered them out to his team of collaborators who was able to identify 69 different protein targets within the human genome, and then take, as I said, 26 or a number of different drugs that had previously been approved by the FDA, and now have 26 of those drugs in trial for repurposing for COVID-19 treatment. And all of this was really enabled through certainly Nevin's work in identifying this interactome, as well as our ability to generate CRISPR at scale in this, what we are now calling engineered cell libraries or ready to assay cells. Because we know not every researcher, not every lab, not every company has the ability to, or the setups to transfect and perform high throughput CRISPR cell editing. We take that out of their hands now and are able to provide those edited cells that are very much ready to assay and run in 96 well plate formats or even 384 well plate formats. So this is one of our newest products that we've recently are in the process of launching, that provides these cell populations cryopreserved. And they simply require a thaw, recovery, and then can be utilized in any number of arrayed screening formats. And with this, we see roughly an average 84% knockout or loss of function in that efficiency within these different populations. So this is a very new and high throughput way to assess loss of function targets. That I like this story quite a bit, and how this connects both of our platforms, going from that Halo capability of guide RNA production into what is our Eclipse platform for industrialized CRISPR cell generation, really, which ultimately we leverage for largely for making clonal populations. And this particular platform does combine, of course, the biological component of working with hundreds of different cell lines, machine learning capabilities to improve our editing processes and expand them to encompass additional cell lines, and of course automation, so that we can transfect and deliver hundreds and thousands of edited cell populations to researchers globally throughout the course of the year. And we enable this with immortalized cell lines as well as induced pluripotent stem cells, enabling both knockouts and knock-ins, depending on the type of edit or research that individuals are performing. So much of our machine learning in how we improve our processes is based on what we call closed loop CRISPR, which allows us to become more predictive in biology and understanding the results that we will see. So this simple graphics really highlight how we are creating our edits, collecting our results, and if we need to, we feed that information back into our systems so that we can improve our editing processes, selecting new conditions, refining the automated editing components or cell handling tools that we use, and continuously improving this workflow to yield more confidence, provide you higher quality results, and ensure that any variabilities in experiments do not come from the tools and the cells that you're working with. So we've seen over time that this process enables us to increase our editing efficiency and ultimately the functionality of the genes, or change in functionality that you as our customers and researchers are looking to achieve. And so through this process, we have assessed 200 distinct conditions for transfection or for nucleoaffecting cell populations. We have done so in over 600 different cell lines. And in totality, we have completed over 250,000 edits, which is really a substantial amount compared to most any other organization that may be leveraging CRISPR capabilities. And what this has enabled us to do, this high level of testing and learning, has allowed us to optimize editing conditions for virtually every tissue type within the human body, as you can see represented by a panel of different cell lines from each of these tissue types. So a handful of breast cancer lines, lung cancer lines, even our challenging immune cell lines, we have identified conditions for being able to create edited populations in. Now, of course, not everything is easy, and not everything will always yield 80 to 90% results. But if we are able to achieve over 50% in a pooled population, we are often quite confident in the loss of function there. And even with knock-ins, when we are able to see just 10 or 15%, we have very strong confidence in our clonal isolation capabilities, and I will explain to you why that is the case, or walk you through that process. But first, I want to give just an overview of this industrialized CRISPR gene editing process that we use. Because really, when we view CRISPR editing, we don't view this as a research experiment to create a cell line. It is a manufacturing process. And everything begins with a technical or feasibility assessment of the edit that's required, whether this be a gene knockout or introduction of a point mutation or a tag insertion. Once we complete that, we go through our standard guide design. Our guides are synthesized on the Halo platform. As that is being done, we prepare our cells for transfection, and we use high throughput nucleofection based tools to enable and perform all of the transfections that we do. Once we've hit that point, we analyze that indel frequency, or in the case of a knockout, the knockout score, to determine how well our transfections have gone and what that population looks like. This then helps us to assess the number of clones that we will need to screen for clonal isolation should we proceed to clonal isolation. Because at this point, many researchers will simply choose to take that pool. And this is that beginning story that I shared with you for the biotech company. They simply decided to utilize these knockout cell pools for their goals and get data much more quickly. Many customers and many researchers will prefer to go the route of having single cell clones generated for them, and we have an entire process that is, again, automated and very, very detailed to ensure that we are delivering the correct clones to you. So if we look at that process a little bit more in detail, what you can see is through that transfection and that genotyping component or module before we proceed to single cell cloning, is everything that we do is done in a high throughput plated format. So we prepare our guide RNA with our Cas9 protein. We make multiple plates so that we have replicates, one for transfection, one as a secondary transfection process that is also for generation of retains, so that we can do mycoplasma testing and cryopreservation testing. We also create a third replicate of this to enable genotyping, so we have multiple batches of the same edits going through for different purposes. Once we've performed our genotyping process, we can then either validate and deliver those edited cell pools, or we take them through our clonal isolation workflow, which will go from 96 well plates into a single cell isolation workflow, or a cell printing workflow. And this is generated out in multiple 384 well plates that are run through a very much automated imaging, refeeding and incubation process that repeats until we are ready to identify potential clones for screening. Again, all of this is done in 384 well plates. All of this is done through automation to ensure that we have minimal human variability introduced. And when we have identified clones, we then pick those clones and plate them out into replicate plates. One that enables us to go through a secondary genotyping and confirm that we do have a pure clonal population, and one plate that is ultimately then utilized for that expansion of desired clones, the final QC and validation and delivery of ultimately product and clonal cell lines to you. So as we look a little bit more in detail of that automated refeeding, imaging workflow, you can see that as we go through each of these plates, we do in fact image 2 hours post plating of these clones. 2 days, 7 days, 9 days. We are imaging over the course of multiple hours to identify single cell populations and ensure that we aren't getting multi-clonal populations here. And you kind of simply look at the live, or the full 384 well plate, as well as a sub corner of that, to show indeed the outgrowths that we are monitoring as we go through that hit picking and clonal expansion. All of this, again, is done to ensure the highest quality and confidence for you of what you are receiving from this tool of gene-edited cell lines. And we found that our edits are very consistent whether we are editing with the same guide sequence 3 weeks apart or 3 months apart, we see the same sequencing or frequency profiles or indel profiles within these populations. And you can see just from technical replit kits the consistency in editing efficiencies in this case. To look at this in perhaps a more interesting depiction of the predictability of the consistency, we've looked at editing with the same guide in the same context, but in different cell lines and at different times. And we see the same indel patterns occurring at the same rates. So this data, I think, really highlights how tight and how well done our process controls are, so that we can become more predictable in our understanding of biology and minimize the amount of technical variability, regardless if you order an edit today or the same edit from 2 years from now. We would anticipate that being the exact same or very much the same in terms of these profiles and ultimately performance. And then moving into just a final summary of the immortalized cell lines, you can see that with just a few handful select lines that we've highlighted, the consistency in high indel frequencies, whether that be workhorse lines such as HEC293 or more specific models such as HepG2 or T-cell alternatives as JERCAS, or even some of our mouse cell lines. And our average editing efficiency in these knockout pools across 130 unique lines is actually quite high at 80% across 490 pools as indicated in this data set. Now, this long tail, you might wonder, what are these particular lower outliers? These often are a result of very challenging cell lines or even essential genes. So it's to be expected that we might have some outliers that have low performance, but we want to show that very clearly that median is quite high, around 80%. Now, I do want to just touch briefly on iPS cells and then a few examples of how we've seen researchers leverage iPSCs as well as this CRISPR at scale for some specific disease modeling work. So we know that induced pluripotent stem cells as a model really enable us to study human biology quite a bit as it's better as we would see in a normal physiological state, because these are not immortalized cancer lines. And what they allow us to do ultimately is take the nascent or the immature and pluripotent stem cell, create a CRISPR edit, either a point mutation, a knockout, and then ultimately differentiate those populations into our cell type of interest. So giving much more insights into true physiological behaviors or responses for disease modeling, regenerative medicine, and developmental biology. And we have been working with multiple iPS lines for several years now, and we actually see even higher editing efficiencies on average with the induced pluripotent stem cells at over 90% from a knockout perspective or loss of function perspective. And even when we look at knock-ins or insertions, we have quite a high editing efficiency here. Excuse me. Of over 90% when we are assessing some small tags. And in this case, we see the performance of knock-ins of HA, 3X flag tags, and a number of others, excuse me, with such high editing scores around that average of 90%. Now, this is just sequencing data, but if we look at our fluorescence or expression, we also see that in this format with our HA tag, we can overlay the expression of the HA tag with our stem cell surface marker SSEA4, as well as DAPI to identify our live cell population. And from this image, these images, you can certainly see that, and in line with our bright field, that virtually every cell within this viewpoint contains that tag of interest. So it is really remarkable to see such high knock-in efficiencies with this type of population, but it gives such a confidence into the editing capabilities that we've been able to generate. So for the last few slides, I simply want to go through a few case studies in which we've had large research groups leverage our capabilities or leverage the capabilities of both these induced pluripotent stem cells as well as CRISPR gene editing. And in our first case, we were looking at TGen, which is the Translational Genomics Research Institute. They are based in Arizona here in the United States, and an affiliate of the City of Hope, which is in Los Angeles, California. And this group is specifically using iPS cells to investigate rare childhood disorders. Their focus here is based on some very intriguing numbers where greater than 25 million Americans are suffering from rare diseases, and there's not a substantial amount of research dollars invested in this. 7,000 rare diseases exist and are affecting millions of people globally. 50% of the patients are affected by rare diseases are often children, which speaks to the mission of TGen and some of their goals, at least within this group. So what they were looking to do is perform some studies, identify genetic factors causing these rare diseases in children, to help guide novel diagnosis and treatment strategies. So they had gone through this process and identified a few targets of interest that they wanted to study, which were largely mitochondrial diseases. But when they began, they wanted to, I should say, they wanted to expand beyond mitochondrial disease and study a number of different rare diseases, but they felt that they were constrained by several challenges. Some of those challenges would include that they believed in editing or creating CRISPR edits in induced pluripotent stem cells was challenging, and that they did not have the technical prowess or capability to learn this and really expand it broadly to create dozens or hundreds of cell lines. They also had limited bandwidth. They did not have the researchers or the people in the lab to dedicate to doing this type of work or even scaling up to making dozens of edits. And then while they had considered exploring outsourcing options and partnering with a CRO to create cell models for them, they felt they would be restricted based on costs and that this would be too expensive and cumbersome for them to do. Now, when they were in conversations with our team at Synthego, they indicated that they could only study 5 targets specifically for mitochondrial disease, and they had narrowed these down based on these presumed risks or limitations of price and bandwidth, and wanted to go with 5 targets that they thought would be the most impactful for their research. As we explained our capabilities with editing dozens, hundreds, and thousands of targets at a time through industrialized CRISPR and our ECLIPSE platform, at the cost that we were able to do, they quickly realized and learned that they could expand their scope of research to cover 60 targets for their pilot experiment across multiple rare diseases and obtain all of these models in just 6 months' time to then perform the analysis on their results, generate data to submit for additional grants, and then certainly come back for subsequent experiments and expanded target lists. So Matt Huntelman, who is the professor of neurogenomics at TGen, really found that, as you can see from this quote, a high throughput standardized genome engineering platform or CRISPR platform enables scaling functional genomics approaches and their eventual adoption for diagnostic and therapeutics pipeline development. So Matt has continued to work with us since this time and expanded beyond those initial 60 targets. My last example in this case is another very large scale program leveraging induced pluripotent stem cells, specifically as they relate to models for Alzheimer's disease and related dementias. Out of or within Alzheimer's, there have been at least approximately 50 genes identified that are associated with Alzheimer's and these dementias. But the interaction or the interplay of these genes is not very clear. So NIH developed the INDY project, which is the Induced Pluripotent Stem Cell Neurodegenerative Disorder Initiative. And they developed this project with a goal of generating a standard collection of hundreds of isogenic iPS lines that carry various Alzheimer's disease or other neurodegenerative relevant mutations, and would be a source for global researchers. So the idea was to have a healthier control cell line and then create multiple different point mutations and various models for each of these gene targets, so that these could be used to identify gene linkages for that are key for developing single drug treatments within these specific diseases and for these specific mutations. So when the INDY group came to us, they presented the idea of generating models for 22 gene targets with 12 variants per gene. And ultimately, what we were able to do through our industrialized CRISPR capabilities was provide them with 264 distinct clones for these different combinations in just 5 months time frame. These various iPS lines now as a bank for the NIH for Alzheimer's and other neurodegenerative diseases can be used for organoid models, vesicular transport, single cell RNA sequencing, whole cell proteomics, and a number of different high content microscopy assays after these cells are differentiated into neuronal populations. So this has become a wealth of information and a huge resource for neurodegenerative researchers globally to leverage, and not something that would have otherwise been possible without an industrialized CRISPR platform. So in summary, I just want to come back to state that at Synthico, we really are solely focused on creating better platforms, models, and tools for genome engineering and leveraging genetics within a myriad of different research applications, whether that be in basic academic research, drug discovery, or really cell therapy development. And we have focused our experience and time to become experts at this editing, producing, as I mentioned earlier, over 250,000 distinct edits with an average editing efficiency of greater than 80%. We have become, in the time as an organization, have become featured in over 950 peer-reviewed journal publications, and are being utilized by researchers throughout the globe in over 45 different countries. And some of our collaborators include these organizations, just to name a few others. And I hope in the future we'll continue to expand into the Korean market through our partners at Philly Korea. So we aim to be your partner in research along this entire process, and give you confidence in your target discovery. If you have plans to move into clinical applications, giving seamless transitions from that discovery to clinical state, providing reliable disease models, and ultimately accelerating cell therapies. And lastly, these are just a recap of the different options that you can utilize gene editing through your experiments and workflows. And I hope that we can continue to be a part of your conversation. And with that, I would just like to say thank you. And again, my name is Rachel, and one of my colleagues who some of you may work with from time to time is Jason Meyer. He was not able to join today, but Jason and I are both readily available to support you as needed, and I'm happy to answer any questions that I can at this time.